Suspended animation is the slowing or stopping of life processes by exogenous or endogenous means without termination. Breathing, heartbeat, and other involuntary functions may still occur, but they can only be detected by artificial means. Extreme cold can be used to precipitate the slowing of an individual's functions. Use of this process has led to the developing science of cryonics. Cryonics is another method of life preservation, but it cryopreserves organisms using liquid nitrogen that will preserve the organism until reanimation. 2-celled embryos have been kept in suspended animation for as long as 13 years. Placing astronauts in suspended animation has been proposed as one way for an individual to reach the end of an interstellar or intergalactic journey, avoiding the necessity for a gigantic generation ship. Occasionally the two concepts have been combined, with generations of caretakers supervising a large population of frozen passengers. Since the 1970s, induced hypothermia has been performed for some open-heart surgeries as an alternative to heart-lung machines. Hypothermia, however, provides only a limited amount of time in which to operate, and there is a risk of tissue and brain damage for prolonged periods. Temperature-induced. In June 2005, scientists at the University of Pittsburgh's Safar Center for Resuscitation Research announced they had managed to place dogs in suspended animation and bring them back to life, most of them without brain damage, by draining the blood out of the dogs' bodies and injecting a low-temperature solution into their circulatory systems, which in turn keeps the bodies alive in stasis. After 3 hours of being clinically dead, the dog's blood was returned to their circulatory systems, and the animals were revived by delivering an electric shock to their hearts. The heart started pumping the blood around the frozen body, and the dogs were brought back to life. On the 20th of January, 2006, doctors from the Massachusetts General Hospital in Boston announced they had placed pigs in suspended animation with a similar technique. The pigs were anesthetized and major blood loss was induced, along with simulated, via scalpel, severe injuries, e.g. a punctured aorta, as might happen in a car accident or shooting. After the pigs lost about half their blood, the remaining blood was replaced with a chilled saline solution. As the body temperature reached 10 C, 50 F, the damaged blood vessel were repaired, and the blood was returned. The method was tested 200 times, with a 90% success rate. Chemically induced. An article in the 22nd of April, 2005 issue of the scientific journal Science reports success towards inducing suspended animation, like hypothermia in mice. The findings are significant, as mice do not hibernate in nature. The laboratory of Mark Pieroth at the Fred Hutchinson Cancer Research Center in Seattle, Washington, placed the mice in a chamber containing ATP-PM hydrogen sulfide for a duration of 6 hours. The core body temperature of the mice dropped to 13 degrees Celsius, and metabolism, as assayed by carbon dioxide production and oxygen use, decreased tenfold. They also induced hypoxia on nematode embryos and zebrafish embryos, placing them in suspended animation for hours, and then reanimating them simply by returning the oxygen to the embryos. Massachusetts General Hospital in Boston announced they had been able to hibernate mice using the same method. Their heart rate was slowed down from 500 to 200 beats per minute, respiration fell from 120 to 25 breaths per minute, and body temperature dropped to 30 C, natural, 39 C. After 2 hours of breathing air without hydrogen sulfide, the mice returned to normal. Further studies are needed to see if the gas had damaging effects on the brain, considering the effect of hydrogen sulfide on the body is similar to hydrogen cyanide. It does not slow the metabolic rate, but rather inhibits the transfer of energy within the cell via ATP. Experiments on sedated sheep and partially ventilated anesthetized pigs have been unsuccessful, suggesting that application to large mammals may not be feasible. In any case, long-term suspended animation has not been attempted. Human hibernation. Therapeutic hypothermia. There are many research projects currently investigating how to achieve induced hibernation in humans. This ability to hibernate humans would be useful for a number of reasons, such as saving the lives of seriously ill or injured people by temporarily putting them in a state of hibernation until treatment can be given. Actual and anecdotal cases of suspected human hibernation or states similar to hibernation exist in the literature. Anna Ekenholm, a Swedish radiologist, who survived 40 minutes under ice in frozen lake in state of cardiac arrest, and survived with no brain damage in 1999. Mitsutaka Uchikoshi, a Japanese man, who survived the cold for 24 days in 2006, without food or water, when he fell into a state similar to hibernation. Polly Hynek, who at age 2 survived several hours of hypothermia-induced cardiac arrest, and whose body temperature reached 64 F. Armando Socaris Ramirez, and a number of other WHOL stowaway flight survivors. As of 2012, only one unpublicized experimental study, undertaken by a private commercial venture, the Dive Lab, has attempted to trigger this state in a human, an expert freediver with a noted ability to downregulate metabolism and brain temperature during diving. Though a switch to trigger this state in animals, let alone humans, has remained elusive, Hibernation seemingly managed to induce a sustainable version of this state in this subject using an as of yet unpublished biomimetic technique modeled on animal hibernators and divers. In this state the subject expressed a prompt constellation of classic, animal-like hallmarks, which, critically, included brain cooling, a necessary condition for expressing any deep form of this state. During the initial exploratory phase of this study, at a university hospital in Sweden, the subject's brain temperature was purported to have plummeted by as much as Ca4C in as little as 15 minutes with no report of shivering. Temperature was measured non-invasively using magnetic resonance spectroscopy. It remains unknown whether subsequent experiments to dimmer metabolism and temperatures to its nadir, or to sustain it for a period longer than about 15 minutes were carried, or whether these may have resulted in so-called suspended animation. Follow-up experiments are supposedly planned for 2014.